Hello and welcome everyone to my YouTube channel. This is Dr. Vijaya Chatterjee. Today's topic is genetic code and Wobble hypothesis. It's a part of molecular biology lecture series. Let's proceed. The central dogma. It shows the flow of genetic information. The genetic information flows from DNA to RNA by transcription and from RNA to protein by translation. So, the central dogma of molecular biology is that proteins are the end products of the information pathway DNA to RNA and RNA to protein. Protein synthesis is called translation because it is this process the four-letter alphabets of DNA and RNA are translated into 21 letters of amino acids of proteins. In other words, the language of nucleic acids is translated into the language of amino acids. Every protein has got a unique amino acid sequence. Information about its amino acid sequence is present in a gene. A gene consists of specific sequence of nucleotides in DNA. A sequence of 3 nucleotides on the mRNA makes a triplet codon. So, these sequences of 3 nucleotides bases in mRNA that code for the amino acid required for protein synthesis are called codons or the genetic code. Triplet codons. George Gamow, a physicist, proposed that if 4 bases code for 20 amino acids, the genetic code must be a combination of 3 bases, a triplet. Example, UUU codes for phenylalanine, AUG, a triplet again, codes for methionine. And this is how it is placed. AUG is one codon, CAU, and so on. Now we will take a look at the logical explanation to why it was most appropriate to consider that a codon is a triplet codon. We know that the DNA and RNA are made up of nitrogen bases, and there are 4 nitrogen bases, chiefly adenine, guanine, cytosine and thymine, and in mRNA thymine is replaced by uracil. So if one base pair represents one codon, then only 4 amino acids are possible. If 2 nitrogen bases form a codon, 16 amino acids are possible. If 3 nitrogen bases form a codon, then 64 different amino acids are possible. And if a codon is quadruplet or pentuplet, then many more other amino acids are possible as shown here. So nature wouldn't want to waste, and so this kind of flamboyance it will not do, and therefore only the triplet code really looks feasible, as it was known that there are 20 amino acids. So having a triplet codon seemed most feasible. Features of genetic code. So the genetic code is non-overlapping, non-punctuated, unambiguous, universal and degenerate. We shall look into each of these individually. So let us proceed. Genetic code is non-overlapping. Codes are read one after the other. The genetic code is read on mRNA from 5' to 3' direction in a continuous pattern. So this is the diagram where the genetic code is read in a continuous pattern. Genetic code is non-punctuated. The codes are always continuous. There are no gaps in between. There is no space. Genetic code is unambiguous. Means a particular codon always codes for the same amino acid. So it is highly specific. It shows specificity. A given codon stands for only one amino acid. Example, UGG always codes for tryptophan. There is no confusion, and this is the diagram where the codes for every amino acid is specific and it holds true every time. For example, AUG stands for methionine without any confusion always. Genetic code is universal. The codons represent the same amino acid in all species. The genetic code is considered to be universal because amino acids are coded by the same codons in genetic material for all living organisms. This indicates that the genetic code has been highly conserved all along the course of evolution. Every protein synthesis begins with an initiator codon and it is always AUG, which codes for amino acid methionine. Every protein synthesis ends with termination codons, which are also known as nonsense codons, because they do not code for any other amino acids. The termination codons or stop codons for nuclear DNA are UAA, UGA, and UAG, and the termination codons for mitochondrial DNA are AGG and AGA. Exceptions to genetic code. We just discussed that the genetic code is highly conserved in all species and it codes for single amino acid. However, there are a few minor variations which are shown in this slide. AUA codes for isoleucine in the cytoplasm and methionine in the mitochondria. UGA is a termination codon in the cytoplasm, whereas it codes for tryptophan in mitochondria. AGA and AGG code for arginine in the cytoplasm and they are termination codons in the mitochondria. Genetic code is degenerate. There are 62 codons for 21 amino acids. One amino acid has more than one codon. For example, serine has 6 codons. Glycine has 4 codons. This is called degeneracy or redundancy of the code. This reduces the effect of mutations. Codons that code for same amino acids are called synonyms. Most synonyms differ only at the third base at the 3' end. This is well explained by the Wobble hypothesis, which will be discussed shortly. Let us look at the codon-anticodon pairing. Each codon in the mRNA is recognized by a complementary 3-nucleotide base sequence on a tRNA called the anticodon. The codon-anticodon pairing occurs in an antiparallel direction. That is, the 5' end of mRNA pairs with the 5' end of the mRNA pairs with the 3' end of the tRNA by Watson and Crick base pairing, where A pairs with U and C pairs with G. If the anticodon of tRNA is recognized by only one codon, there should be different tRNAs for each codon present on the mRNA. So you can see here that there is one codon CGA. So if the codon of each tRNA is recognized by only one codon, there should be different tRNA for each codon present on mRNA. However, there exists only around 40 to 45 tRNAs for 62 codons that code for amino acids. Therefore, certain tRNA molecules must be pairing with more than one codon, and this is once again can be better explained by the Wobble hypothesis, which we will discuss. So this is the traditional Watson and Crick pairing, and there is a different kind of pairing that takes place in the between the mRNA and tRNA at the third base pair. Let us look at that. We recently looked at what are the Watson and Crick base pairing. Now let us look at the Wobble base pairing. This is a different kind of base pairing. Wobble base pairing, there are four main kinds. So generally it is seen that at the 5' end of anticodon in tRNA, if there is inosine, it can pair with the 3' end of the codon with uracil, adenine or cytosine, any one. So you can see that there is no, it is not specific, and guanine can also pair with uracil, which is also an unusual base pairing. So this means this is a wobble base pairing, and wobble means to move unsteadily. Phenomenon gives rise to degeneracy of the genetic code. So let us look at this into a detail. This was first discovered by Francis Crick while he was studying codon-anticodon pairing. He discovered that a single tRNA can recognize more than one codon. This is possible because the third base pair in a codon fails to recognize the complementary base pair in anticodon, and therefore the first two bases of mRNA always follow the Watson and Crick base pairing, uh, whereas the third base pair is loosely bound with the corresponding anticodon. So you can see that the first two bases is always following the Watson and Crick rule, whereas the third base pair is loosely bound to whatever is present in the anticodon region. So there is wobbling at the third base pair, which means that the pairing is not as per Watson and Crick's rules. This brings about degeneracy in the genetic code, and this also is highly useful because the degeneracy code prevents mutations, and this would, if there was no degeneracy, that would severely hamper the speed of protein synthesis. Wobble hypothesis. So now we can easily look into the wobble hypothesis after understanding the basic concepts of Watson and Crick pairing and wobble pairing. According to this hypothesis, only the first two bases of the codon in mRNA have a precise pairing with the bases of the anticodon of tRNA, while the pairing between the third base of codon and anticodon may wobble. So wobble means to sway or move unsteadily. Thank you for watching. Like, comment and share, and subscribe to my channel.